Today, I want to dig into some engineering and chemistry. I'm going to give you a pretty deep dive into a set of technologies that we've been designing and deploying for measurements that I think are somewhat neglected and quite complementary to the last talk that you've heard. I will give two vignettes of projects that we've been working on. One to share with you our approach to designing and developing new tools. So I'm a bioengineer and mechanical engineer by training, so that is the focus of the research I'll talk about today. And then also to potentially stimulate questions or thoughts or possible connections between the work that you're pursuing and the work that's happening in my group. So hopefully we'll meet those two goals as we walk through the short presentation today. So what I'm going to talk about is really tool sets to measure cellular to molecular heterogeneity. And for us, that means with precision microfluidics, an area that we've focused on over the past couple of years has really been around, again, this gap that you heard mentioned in the previous talk, but I think could benefit from a tremendous upswell in new high specificity tools. And those are questions that really involve the highly specific measurement of different proteoforms. I'll walk through essentially what that means. So when we think about cellular heterogeneity, we know that cell-to-cell variation is important for disease, understanding disease progression and life processes. We also know that molecular heterogeneity is important. Oftentimes we think about single-cell tools and just the tremendous advances that tools specifically focused on direct measurement of DNA and RNA are bringing, both to our understanding of biology and biomedicine, but also really to the way that we ask questions. I have to say that those tools obviously are quite powerful and have opened new doors and will continue to do so. But I do want to remind you, even beyond what we've been thinking about this morning, that there is an entire constellation of molecules that are out there, proteins, that exist in what I consider to be a combinatorial explosion of different states. So specifically, I'm talking about proteins, their different isoforms, the different site-specific features. Key among these are things like post-translational modifications, of which there are upwards of hundreds, if not thousands, of modifications that occur in biological systems. And then very, very importantly, this combinatorial explosion, as I call it, of a combination of each of these. So the way that the different isoforms, site-specific features, changes in conformation can all be reflected in the specific function of a specific protein molecule as it exists in nature. So when we talk about proteoforms, and there's a really nice set of articles that introduce this more blanket concept, this more umbrella concept, extending beyond the concept of protein isoforms. And the key reference I provide right here for you if you're interested. But when we think about these molecules, we know that to get the selectivity we need, the use of antibody probes is just not going to cut it. And that's because estimates of the number of proteoforms that exist span anywhere from 10 to the 6th to 10 to the 9th different unique molecular species. So even if we could generate affinity reagents for each of those species or species of interest, thinking about generating on that scale, well, potentially possible, I'm not going to wait for it, potentially possible is daunting to say the least. And so I would urge you, if you are thinking about ways to help advance single cell understanding of biology and biomedicine, that thinking about some of these targets that are quite literally blind spots to the tools that exist today, not measurable at the DNA level, not measurable at the mRNA level, and certainly not measurable with many existing of the immunoreagents that are out there. So to quote another really nice article that sort of introduced this umbrella concept of proteoforms, these specific molecular forms are a complex and largely uncharted world of natural proteins. So that for me links when we think about things like the Human Protein Atlas, the Human Cell Atlas, and these different large-scale efforts, which are quite important in making good headway. To hear the words uncharted world really kind of piques my interest, as well as others, certainly, with thinking about how can we understand not just the state, but also the role of these molecules that were these protein molecules that we're not really measuring at all in many cases, with the exception of a tiny important handful. I guess the flip question of that is, of those that we're not measuring, how many of them are important, and we're not seeing them at all. So where I really started with this, again, as an engineer, as a measurement scientist, as an analytical chemist, I had a chance to spend part of my sabbatical in a clinical lab a couple of years ago with Mark Pegram and Stephanie Jeffrey, visiting them periodically at Stanford, and really opened my eyes to how important some of these proteoforms are, not just in understanding, but also in clinical medicine. And this has formed the basis for several projects that we've pursued and are currently pursuing. And this is around questions of oncoproteins and the role that different proteoforms of those oncoproteins play. So again, I am going to use this time with you to deep dive into specific questions and measurement systems, with the hopes that will pique some interest in you and maybe the tools or approaches that you're developing, and you can help us, not just me, but our field, tackle these problems. So cut to the chase, the example that I'm going to use today is the ERBB2 protein. This protein is also known as a HER2 protein. It is a transmembrane protein. So if this is the membrane of the cell, we expect HER2 to span that membrane. There's two important domains for the purposes of today's discussion. One is the extracellular domain, and the second is an intracellular tyrosine kinase domain. This protein is large. It's about 185 kilodaltons. There's really two important things to take away today about the HER2 protein, and then an implication. And the first important thing is this extracellular domain of this 185 kilodalton protein was the very first protein domain that was targeted by the first FDA-approved targeted cancer therapy. So when we're thinking about moving away from chemotherapy and radiation therapy, or augmenting those with a targeted therapy, trastuzumab, a humanized monoclonal antibody, fit the bill, also known as Herceptin, in terms of being able to target that extracellular domain of HER2, which is oftentimes dramatically overexpressed on cancer cells. In addition to understanding the clinical function of this transmembrane protein in that extracellular domain, I want to draw your attention to the fact that even this HER2 protein exists not as a single molecular form, but as an entire family of molecular forms. And this is actually quite common. And so we see what are called truncated isoforms of HER2 playing roles in cancer biology. And we have truncated isoforms that span from 110 kilodaltons or so. So they're called truncated because they're missing that 75 kilodalton extracellular domain region out here. And also